So here's some practice. Try to translate the following here. So AUG, remember how you go. You go A, U, G. First base, second base, third base. So first base across, and the second base, they both meet in this box. And then you bring the third base from the right, and you get MET. Let's do GAG. The first base, which is G. Second base is A. And so both of these meet here, just like a Punnett square. But you need the last base to tell you which one it is. GAG is glue, glutamic acid. Then you have CCA. C, C, A. Then you have ACG. A, C, G. Then you have UAA. U, A, A, which is stop. Practice those. Check for understanding. What do you think these are? Pause the video right now and practice. When you're ready, then you can hear this now. So that's a chromosome, RNA polymerase, DNA, nucleus, or actually here it should be the process of transcription. So let's correct that, transcription. You should have translation there, the whole process of making a protein, DNA, polypeptide chain, codon, anticodon, ribosome, mRNA, tRNA, amino acid, nuclear membrane, nuclear pore, and continuing on. Protein diversity. Remember that there's different types of proteins. Here is a variety of proteins. They do a lot of things in the body. They're enzymes that help you digest. They're enzymes that help you build DNA and other molecules. There are enzymes that help you get energy. There are enzymes that help you move your muscles. There's enzymes for a lot of things. There's a lot of proteins also. Again, enzymes are proteins, but there's other types of proteins too. You have proteins in your eye, in your skin, in your muscles. Proteins practically that reveal all your traits, like your hair color, your eye color, your hair type, your height. Proteins or hormones, also like insulin. And so there are many types of proteins. They're all over the body, and that's what builds our body. That's what gives us our traits. So there's thousands of them. And how are they different? Well, remember first there's 20 different amino acids, and you can combine them in all sorts of patterns and shapes. And some proteins range from 50 to 50,000 amino acids in length. What makes them different again is, first of all, their length, the amino acid order. A lot of amino acids can be repeated in different ways. And when they get together, these proteins have different shapes. Those shapes allow them to become to have different functions. So remember, the amino acid sequence leads to the shape, which leads to the function. Then we have mutations. Remember, the mutations can either be somatic in body cells or a gamete. When they happen in gamete cells, then the whole child will be affected because that makes that's the sperm and the egg which makes the child. So here you can see that the mutation led to every cell in the body. It was carried on to the offspring because it was in the sperm or egg. But if the mutation happens in a somatic cell, so it doesn't happen in the sperm or egg cell stage, it happens when you're making the body or sometime later, such as skin cancer, lung cancer, or other types of mutations that happen, then that mutation will only affect certain parts of the body, but it will not be carried to that offspring. It will not go to your offspring. So again, mutations, they're a source of genetic variety. If they're positive or beneficial mutations, then they help us evolve. Most mutations are harmful. Some are neutral. Remember, they're neutral if the amino acid doesn't change or the protein doesn't change. If the protein still stays in the same order of amino acids, it'll still fold the same way, same protein, and you get lucky. Then you have gene mutations versus chromosome mutations, which we're going to talk about. What are the causes of mutations? A lot of environmental things. Again, like we talked about sun exposure, we talked about cigarette chemicals, and a lot of things in the environment. such as heavy metals and things like that. But they can also just be random as your cell, your DNA replicates, as it copies itself. Sometimes it copies itself because there's so many billions of bases it has to copy all the time, and there's going to be a mutation. And sometimes, or most of the time, your cell fixes it. There are repair enzymes that fix those mutations, but sometimes they go unchecked and could lead to a problem. Here's a gene mutation. They're sometimes called point mutations because they happen at a single base. So you have mutations where, here's an example, there's no mutation, and here you get this amino acid sequence. So pay attention to that carefully. Then we have a substitution. So right here, there should have been a C and a G, but instead we put a T and an A. So we changed one base. And what that does in this case, if the amino acid changes, see, it was supposed to be glue and now it's lyse, now we're going to have maybe a problem, or if we're lucky, we get a beneficial mutation. Then we have a mutation where we actually add something. We actually inserted the C. And so that, in that case, causes a frame shift, because now when you add something, everything shifts over to the right. And that messes up all the codons from there, all the amino acids. See, these 3 and beyond get messed up. And now they're different amino acids. So you're very likely to have a problem here. And then we have deletions, where you take away a base, and then everything slides to the left. So again, that's another frame shift. So remember that insertions and deletions, because they cause a frame shift, they mess up all the amino acids after that point. Those are the worst, insertion and deletion. Substitutions aren't that bad. They are bad because, if they mess up the protein, obviously, but they do affect only one part, and they only affect one codon. Neutral mutations, again, here you have the original sequence of amino acids, and let's say we substitute it, but it still comes out the same. See, this amino acid sequence and this amino acid sequence is the same. It's neutral. It's not going to have an effect. Same protein comes out. Here you have chromosome mutations. Chromosome mutations are different from gene mutations because they affect a bigger area of DNA. They affect a chromosome, which has thousands of genes that are affected there. And here are the following. So review those. Then you have gene expression. Remember that as we're developing from a zygote all the way to an embryo, and then to a fetus, and then to an adult, we're going through the process of mitosis. And all the cells of our body are identical. But then why do we have different cells? Like nerve cells, muscle cells, red blood cells, white blood cells, and like the cells that you see here, for example, that are different in shape and function. Well, the reason is they still have the same DNA, but some genes are on and some are off. So the genes that are necessary for that cell are on, and the genes that are not are off. For example, here in the skin cell, brain, heart, and lung, it has all the DNA, all the same genes. And except in a skin cell, the skin gene would be on, everything else would be off, because you only want that skin protein to be created and not any of the other proteins. And that's how you save energy as a cell. You don't want everything on, because then the cell will be confused. It'll say, okay, am I skin cell, heart cell, muscle cell? What am I? So you want to keep it simple and only have those genes that are on. And because of science, we can convert cells. We can turn a skin cell to a brain cell. How would you do that? You could just turn off the skin gene and turn on the brain gene, and then it'll become a neuron. Genetic engineering. This is a process of taking a cool gene that we like from an organism, take it out, and put it into another organism here for our benefit. A lot of times it's for crops, so that we can make crops that last longer, that have a better yield or produce more, are juicier, are tastier, and other things. Also to help cows produce more milk, healthier milk, and help even cows produce milk that has vitamins in it or medicines in it. Sometimes we also do it just to be fancy, or just to have fun in the lab. Like they were able to get a jellyfish protein or a jellyfish gene into a mouse, so that the mouse can make fluorescent protein or a glowing protein, and so now the mouse glows. What for? Sometimes for fun, but it has some medical uses sometimes. How do you do that? Well, review the steps here. you first have to take the DNA that you like out with a restriction enzyme. Once you get the restriction enzyme, you're going to have sticky ends. You put them together into a plasmid, and then you put the plasmid back into a bacteria here. And then the bacteria will make, in this case, insulin. Here's some practice problems. Translate the following into a protein. In this case, you have RNA. So in this case, you just have one step. Remember, DNA, RNA to protein, so RNA to protein is one step translation. So you can just go ahead, use your codon chart. You're done. Now, in this case, you're going to translate this one, but you start with DNA. Remember, the central dogma goes DNA to RNA to protein. So you first have to transcribe and then translate. So transcribe to RNA, translate to protein. And you're going to have to use your codon chart for these, right?